Hello, we are the iGEM team from the University of Göttingen. Göttingen is a city in Lower Saxony, Germany. In 2004, it had a population of 129,466 inhabitants, of which 24,000 were students. That is 18.5% of the population. The halls of the University of Göttingen have been a host to eminent scientists such as Max Born, Carl Gauss, Otto Hahn, and Max Planck. This year, we are participating in the International Genetically Engineered Machine competition with an interesting project. We intend to develop a novel screening technique for different invasive fungi, such as Aspergillus fumigatus, Candida albicans, and Aspergillus nidulans. The basic idea is to tag the invasive fungal cells with a special and customizable protein marker. Eventually, by modifying the protein marker with a proper signal molecule, radio-labeled amino acids, or chemical moieties, it could not only be applied as a diagnostic tool, but also as a therapeutic strategy. Nowadays, more than 300 million people suffer from fungal infection-related ailments, some of which are as extreme as chronic illness, blindness, and even death. One of the most important human pathogenic fungi is Aspergillus fumigatus. Infections caused by any number of the genus Aspergillus are known as aspergillosis, and Aspergillus fumigatus is known for causing a severe form of this infection in patients with a compromised immune system, in whom it has a mortality rate of 30 to 85%. Early diagnosis is critical for a favorable outcome, but that is difficult to achieve with current methods since fungal detection markers start appearing in body fluids only at advanced stages of the infection. That is, when the patient has a lower chance of survival. Methods such as a latex agglutination test, ELISA, and the measurement of the concentration of 1,3-beta-D-glucan in the plasma only yield results at such advanced stages. Furthermore, tissue samples are not always accessible for histological diagnosis. PCR-based methods has always been the mainstay for the diagnosis of these diseases, and for a good reason. The results are highly specific and can be relied upon at early stages of the infection as well, but they do not provide much information about where the infection is localized to. Even if it did, microscopy has been the most reliable means of establishing whether an individual has a fungal infection or not. Due to the limitations in visually ascertaining which fungus it is, other techniques had to be relied upon. However, we believe that we can enhance the diagnostic capability of microscopy by increasing not only the specificity of the examination, but also the visibility.
A useful strategy may come from the most recent advances in cancer therapeutics, using synthetic small peptides that bind to surface proteins of the cells. Some small peptides are nowadays being used as diagnostic and therapeutic tools for cancer patients, and some others are in development. The basic principle is to find a surface receptor that tumor cells express in higher quantities than healthy tissues and use and modify its natural small peptide ligands. These small peptide ligands are modified from its natural form to increase its stability and half-life in the body, and to include radio-labeled nucleotides that can be used for diagnostic imaging or therapy. Small peptides have many advantages over other molecules like antibodies and antibody fragments. They are small and can be synthesized and modified less expensively. They show high diffusion rates in tissues, rapid pharmacokinetics, and high signal-to-background ratios. They are generally non-immunogenic. They can be modified to be used in SPECT and PET imaging. Those small peptides are usually ligands or peptide hormones that naturally bind to cell receptors, like somatostatin or cholecystokinin peptide. Their structure has been finely shaped over the long course of evolution to bind specifically to its receptor, limiting the available candidates only to those ligand-receptor pairs that occur naturally and reducing the diversity of cells to be identified or treated. But what if we could accelerate that process and generate our own customized synthetic peptides to target whatever surface protein we want? This is possible with the power of high-throughput screenings.
Entering our project, first of all, we work with fungi, not tumor cells. but the underlying principle is essentially the same. We start by selecting a number of surface proteins from Aspergillus fumigatus and other pathogenic fungi and get their gene names and coding sequences from bioinformatic databases. Then we ask specialized laboratories to provide us with the cDNA of those organisms. This cDNA is totally safe to handle in S1 laboratories like ours. Next, we amplify via PCR the genes we are interested in, and after some cloning steps, we transform them into yeast cells. Then we generate a peptide library with millions of different random sequences. These peptides are in the size range of 20 to 30 amino acids. These peptides will be then tested and screened for their ability to bind the surface proteins whose genes we clone inside the yeast cells. The test for this interaction is called a yeast-2 hybrid assay.
This assay is used to detect the interaction between two proteins. As for our project, it was used to screen the interactions between peptides from our library and the surface molecules from different fungi. Yeast exists in two distinct mating types, type alpha and type A. They can fuse to form a single cell with genetic material from both, and this phenomenon can be exploited to study interactions between the molecules in this assay. A transcription factor enables the expression of genes to which it binds. The part that binds to the gene to be activated is called the DNA binding domain, while the part that recruits molecules essential for expression is called the activation domain. In this case, this factor is split into two parts, both of which are required for the gene to be fully active. The protein attached to the DNA binding domain is called the bait, while the part with the activation domain is called the prey. When the bait and prey come together, the upstream activating sequence triggers an otherwise inactive gene that is associated with it. The yeast strains used here are missing a gene that codes for the synthesis of an essential nutrient. In our case, this was the amino acid histidine, meaning these cells could only grow when supplemented with histidine in the growth medium or if the activity of the histidine gene would be restored. Any protein that binds to the bait will activate the transcription of a histidine reporter gene. The first step is to construct a bait plasmid and a library of cDNAs in the prey plasmid. Both plasmids are transformed into two mating types of yeast cells. The A type were placed in tryptophan-deficient medium and the alpha type were placed in leucine-deficient medium. Afterwards, these two medium were mixed so as to let the A and alpha cells mate together. After the mating, the cell growth will appear only in the cells which contain both kinds of plasmid. Plating on agar lacking histidine selects for cells that contain the genes for the interacting proteins. Wherever there is growth, it is an indication of interaction between the two parts of the transcription factor. Binding proteins will be identified by sequencing the DNAs of plasmid isolated from the mated cells.
As promising as all of this looks, it's easier said than done. In fact, we are yet to surmount a few challenges on the way. Firstly, we need to test large quantities of potential peptide candidates to get some positive ones, and secondly, within those positive peptides, we need to identify false positives and weed them out. This is due to the high false positive rate inherent to the yeast-2 hybrid assay used in this project. Finally, even when we find one or more promising candidates, the way to get a functional drug or diagnostic tool is far from complete. But someone has to take the first step. In a world of relentless competition, novel methods of production and their products are valued immensely. We propose a novel pipeline to generate synthetic and customizable biological products for diagnostics and therapy. We are still at the very first proof of principle, but we feel much honored to contribute with our effort.